Hello there, and welcome. I'm so glad you found your way here tonight. Maybe you've had a long day, or perhaps you're simply the kind of person who likes drifting off to the whisper of ocean stories rather than counting sheep. Either way, you've chosen the perfect journey. There's nothing you need to do but rest. Let your mind grow calm, your breathing slow and steady, and your body settle into the comfort beneath you, as if the ocean itself is cradling you in its gentle currents. Because tonight we aren't simply looking at coral. We're drifting down through clear tropical waters, descending slowly past schools of silver fish, past swaying beds of seagrass, down to where the reef transforms into something magical, down to where the coral forest begins, and the sea fans spread their delicate arms into the current like living lace against the blue. Sea fans are among the ocean's most elegant architects. They build not with stone or rigid calcium like their stony coral cousins, but with flexible organic frameworks that move with the water, that bend and sway and dance in currents that have flowed the same paths for thousands of years. These are not solitary creatures. Each fan you see spreading its intricate branches across the reef is actually a colony of hundreds of thousands of tiny animals working together, breathing together, feeding together, as one living sculpture. They're called gorgonians, named for the gorgonian that forms their skeleton, that remarkable protein that gives them both strength and flexibility. And tonight, we'll explore their world slowly, gently, the way you might walk through a forest on a quiet afternoon, noticing each detail, each small wonder, as sleep finds you somewhere along the path. If you enjoy these ocean journeys, consider subscribing so you don't miss the next one. And if you're still awake enough, let me know where in the world you're listening from. Now let your eyes grow heavy. Let the day fall away, and let's descend together into the coral forest, where time moves differently, where growth is measured not in days but in decades, and where patience is written into every branch. When you first encounter a sea fan underwater, your breath catches, not from lack of air, but from beauty. There, anchored to a rocky outcrop, or the skeletal remains of ancient coral, rises a structure so intricate, so perfectly geometric, that it seems impossible it grew rather than being designed. The fan spreads in a single plane, its branches forming a lattice of such delicate precision that light filters through it in patterns that shift with every pulse of the current. Purple perhaps, or yellow, maybe a soft orange that glows when sunlight angles through the water at just the right slant, or a deep wine red that makes the whole structure look like it was carved from a single precious gemstone. But step closer, kneel in the sand and look carefully. What appears from a distance to be a smooth surface reveals itself as something far more complex. The branches aren't smooth at all. They're covered with tiny bumps, small raised calyces where individual polyps live. Each one a miniature anemone, each one with 8 delicate tentacles that it extends into the current when the time is right for feeding. There are hundreds of them, thousands, sometimes hundreds of thousands of these tiny creatures, each no larger than the head of a pin, each one genetically identical to all its neighbors because they all grew from the same founding polyp that settled here years or decades ago. The structure itself is a marvel of biological engineering. Deep inside, running through the center of every branch, like the trunk of a tree or the bones of a body, lies the axial skeleton. In many species, this skeleton is made primarily of gorgonin, that unique scleroprotein that gives sea fans their characteristic flexibility. It's somewhat like collagen, the protein that makes your own skin elastic and your tendons strong. But gorgonin has its own special properties. It's horn-like, slightly translucent when you examine it closely, and it has exactly the right balance of rigidity and bend. Enough structure to support the colony as it grows upward and outward into the water. Enough flexibility to move with the surge and swell without snapping. In some species, this gorgonin skeleton is reinforced with calcite, the same mineral that makes up limestone and the shells of snails. These calcite structures, called sclerites, are tiny, microscopic really. They're embedded throughout the living tissue like chainmail woven into cloth, providing additional protection and support. Under a microscope, they're beautiful in their own right, each species producing sclerites of characteristic shapes. Some look like dumbbells, others like tiny clubs or needles or stars. Scientists use these shapes to identify different species of sea fans, because even when the living tissue is gone, even when a sea fan has been dead for years and all that remains is a dried skeleton on a beach somewhere, these sclerites remain, telling the story of what once lived there. The living tissue covers this skeleton-like skin. It's called conenzyme, this layer of connected flesh that unites all the polyps into one functional organism. Within this tissue run channels, tiny tubes that connect every polyp to every other polyp in the colony. Through these channels flows nutrition, chemical signals, perhaps even something like communication, though we can only guess at what information might pass between polyps. When one polyp captures food, the nutrients don't stay isolated. They spread throughout the colony, shared among thousands of individuals. When danger threatens one part of the fan, chemical warnings might ripple outward, causing polyps elsewhere to retract their tentacles, to pull themselves into the safety of their calyces. The colony functions as a single superorganism, more like a forest of connected trees sharing resources through underground fungal networks than like a city of independent individuals. The colors, those gorgeous hues that make sea fans so photogenic, so beloved by divers and underwater photographers, come from different sources depending on the species. In shallow water sea fans, much of the color comes from symbiotic algae called zooxanthellae. These single-celled dinoflagellates live inside the tissue, sometimes more than 1 million of them, in a single square centimeter of coral flesh. They're photosynthetic, which means they capture sunlight and turn it into energy, producing oxygen and organic compounds that the coral polyps can use. In return, the polyps provide the algae with a safe home, with nutrients from their waste products, and with access to sunlight. The algae are typically brownish or greenish, but their presence can contribute to the overall coloration of the fan. When you see a sea fan that's a rich brown or olive color, you're seeing the color of millions of tiny algae shimmering through translucent coral tissue. But many sea fans produce their own pigments as well. These are often carotenoids, the same family of compounds that makes carrots orange and tomatoes red. The exact shade varies from species to species, from individual to individual, and sometimes even from one part of a colony to another depending on light exposure or water chemistry. A sea fan growing in the bright shallow reef might be a different color than its genetic twin growing deeper, in the dimmer blue light where only certain wavelengths penetrate. The purple in purple sea fans, that distinctive hue that gives Gorgonia ventellina its common name, comes from specific pigments the coral produces itself, compounds that may help protect its tissue from too much sunlight or from other environmental stresses. And the shape, the characteristic fan shape that gives these corals their name. It's not random, it's not decorative, it's functional. Evolved over millions of years to solve a specific problem that every filter-feeding organism in the ocean must face. How do you maximize your contact with moving water when you can't move yourself? Sea fans solve this problem beautifully. They grow in a single flat plane, spreading their branches not in all directions like a bush or tree, but in one plane like a Japanese hand fan. And they orient this plane perpendicular to the prevailing current. Imagine opening your hands and holding them flat against a breeze. That's what a sea fan does. The water flows through the lattice of branches, and as it does, it brings food, plankton, tiny particles of detritus, nutrients dissolved in the water itself. The polyps extend their tentacles into this flowing cafeteria, this endless conveyor belt of food, and they catch what they can. The spacing of the branches, the size of the gaps in the lattice, these too are not random. They're optimized, too close together, and the water flow slows too much, creating areas of still water where food settles rather than being presented to polyps, too far apart, and too much water rushes through without being filtered effectively. The ideal spacing varies depending on the typical current speed in the environment where the sea fan lives. A sea fan in a high-energy environment with strong, constant currents might have branches spaced further apart. One in a calmer area might have a tighter mesh. Over evolutionary time, over thousands of generations, the architecture has been refined, not by conscious design, but by simple survival. The colonies that happened to grow with more optimal spacing captured more food, grew larger, produced more larvae, and passed on their genetic instructions for that particular branching pattern. Growth, when you're a sea fan, is not a hurried affair. There are no growth spurts, no rapid expansions, just the slow, patient accumulation of new polyps, new branches, new layers of skeleton deposited season after season, year after year, decade after decade. In the warm, shallow waters of the Caribbean, where food is abundant and sunlight feeds the symbiotic algae, a sea fan might add a few centimeters of branch length in a year, perhaps 5 centimeters, perhaps 10 in an especially good year. In cooler waters, or at greater depths where food is sparser and light dims to a perpetual twilight, growth slows further. A centimeter a year, half a centimeter, sometimes even less. The factors that influence growth are many and interconnected in ways we're still working to understand fully. Water temperature plays a crucial role, affecting metabolic rates and the speed of biochemical processes. Warmer water, up to a point, generally means faster growth because chemical reactions proceed more quickly, because the polyps can process food more efficiently, because new tissue can be built at a higher rate. But there are limits. Too warm, and the benefits reverse. Stress increases. Energy must be diverted to coping with heat, rather than being available for growth. The coral might stop growing entirely, waiting for conditions to improve. Food availability is perhaps even more important than temperature in many environments. A sea fan in nutrient-rich water, where plankton blooms regularly, might grow twice as fast as a genetically identical colony in clearer, more oligotrophic water. The relationship between feeding and growth is direct in species without symbiotic algae. More food captured equals more energy available for building new tissue and new skeleton. In species with zooxanthellae, the relationship becomes more complex. The algae can provide baseline nutrition, but additional feeding might still be necessary for optimal growth, particularly during reproduction when energy demands spike. Light matters tremendously for species with symbiotic algae. These zooxanthellae need light for photosynthesis, and the amount of light they receive directly affects how much energy they can produce for their coral host. A sea fan growing in bright shallow water might photosynthesize enough energy to meet most of its needs, supplemented by occasional feeding. The same species at depth, where light intensity is reduced to perhaps 10 or 20% of surface levels, must rely much more heavily on captured plankton because its algae simply can't photosynthesize as efficiently in dim light. This is part of why depth gradients often show declining coral growth rates as you descend, even when other conditions remain similar. Current speed and consistency affect growth in subtle ways. Strong, steady currents bring food reliably and remove waste products efficiently, both beneficial for growth. But excessive current creates mechanical stress. The colony must invest more energy in building stronger skeleton to resist being torn from its holdfast or having branches snapped. Extremely turbulent flow might prevent polyps from extending fully to feed, reducing food capture and thus available energy. The sweet spot varies by species, but most sea fans seem to grow best in moderate, relatively steady currents. Think about what this means. That large purple sea fan you see on a Caribbean reef, the one spreading its branches 1.5 meters wide, the one that seems to dominate its patch of reef like an ancient tree in a clearing, that fan might be decades old, 50 years, 70 years. Some estimates suggest that the largest individuals of certain species could be a century old or more, though aging sea fans precisely is difficult. They don't lay down annual growth rings the way trees do, at least not in the same obvious way. Some species do produce growth bands in their skeletal axis that can be counted, but these bands don't always correspond neatly to annual cycles. Researchers trying to determine the age of a sea fan must sometimes use indirect methods. They might measure the growth rate of marked colonies over several years, then extrapolate backward based on the size of larger colonies. Or they might look at the chemistry of the skeleton, seeking signatures that correlate with known environmental events. The chemistry of growth is itself fascinating. As the coral builds new skeleton, it incorporates elements from the surrounding seawater in proportions that vary slightly depending on environmental conditions. The ratio of certain trace elements, or the isotopic composition of carbon and oxygen in the skeleton, can vary with water temperature, with pH, with the availability of different nutrients. These chemical signatures become locked into the skeleton as it forms, creating a permanent record. Scientists can analyze the chemistry along a cross-section of the skeletal axis, moving from the outer newest growth toward the center where the colony first began, and read backward through time, seeing how conditions changed over the life of the coral. The pink sea fans of British waters grow even more slowly, about 1 centimeter per year, perhaps a bit more under ideal conditions, which means that a fan 30 centimeters tall could be 30 years old. It could have begun its life when the current generation's parents were still children themselves. These slow growth rates are typical of many gorgonians, especially those in temperate or deep waters, and this slowness has profound implications. It means that recovery from damage is measured not in months or even years, but in decades. A sea fan broken by a careless anchor or crushed by fishing gear doesn't regrow quickly. The colony might survive, might slowly regenerate damaged branches over years, but it will never be the same. The lost time, the lost growth, that's gone forever. In the deepest parts of the ocean, where some species of Gorgonian corals create forests in the cold darkness thousands of meters down, growth becomes almost unimaginably slow. Some deep-sea Gorgonians add less than a millimeter of growth per year, less than the thickness of a fingernail. There are bamboo corals and gold corals from the deep Pacific that have been radiocarbon dated to be thousands of years old. One specimen of the gold coral, Gerardia, was determined to be more than 2,700 years old. It was already ancient when Rome was founded. It was growing in the deep sea while the pyramids were still being built in Egypt. Another deep-water species, a black coral called Leopathis, has been found with specimens over 4,000 years old. These are among the oldest living organisms on Earth, rivaling the ancient bristlecone pines that cling to mountainsides in California and Nevada. But even these extreme ages pale compared to some estimates. There are suggestions, though not yet confirmed with certainty, that some of the largest deep-sea Gorgonian colonies could be even older, 6,000 years, 8,000 years. If true, these would be organisms that began their lives before the dawn of recorded human history, before the invention of writing, before the first cities rose in Mesopotamia. They would have been growing, slowly adding polyp after polyp, branch after branch, while humans were still hunter-gatherers, while the Ice Age was ending, and sea levels were rising, flooding coastal valleys, and creating the modern coastlines we know today. The growth process itself is both simple and complex. It begins with a single polyp. This founding polyp settled here as a larva, perhaps weeks or months ago. It attached to a hard surface, a rock or a dead coral skeleton, or even a shipwreck, and it underwent metamorphosis, transforming from a swimming planular larva into a sessile polyp. It secreted its first bit of skeleton beneath itself, creating a tiny holdfast that glued it firmly to the substrate. And then it began to grow upward, the base of its body gradually building the beginnings of the axial rod that would one day support a colony of thousands. But a single polyp does not make a sea fan. Growth requires multiplication, and this happens through asexual budding. The founding polyp begins to produce buds, small outgrowths from its body that develop into new polyps. These new polyps remain connected to the parent, sharing the same skeleton, the same tissue, the same circulatory system, such as it is. Each of these new polyps can then produce buds of its own, and those buds can produce more buds, and gradually, through this process of branching and rebranching, the colony takes shape. The main axis extends upward. Side branches form, spreading left and right in the plane of the fan. Those side branches themselves branch, creating the secondary and tertiary network that forms the characteristic lattice. All of this happens slowly. A branch might extend a few millimeters in a month. The addition of a new polyp might take days or weeks. The skeleton grows along with the living tissue. The polyps continuously secrete new gorgonin, adding layer after layer to the axial core. In species that incorporate calcite, new sclerites are produced and embedded in the tissue. The colony doesn't grow uniformly. The tips of branches, where the newest polyps are located, grow most actively. Older parts of the colony, closer to the base, grow more slowly, if at all. Their role now structural rather than expansive, and damage can alter growth patterns. If a branch breaks, the colony doesn't simply extend that branch back to its original length. Instead, it might divert growth resources elsewhere, developing new branches from points below the break, gradually filling in the damaged area with new growth that might not follow quite the same pattern as the original branches. Environmental conditions modulate growth continuously. A year with abundant plankton might be a good year for growth. A year with water that's slightly too warm or too cold might slow things down. Storms can damage colonies, breaking branches, sometimes even dislodging entire fans from their holdfasts. But they can also stimulate growth in a strange way. A fragment broken from a parent colony, if it lands on suitable substrate and manages to attach, can become the founder of an entirely new colony. This is asexual reproduction through fragmentation, and for some species in some environments it might be more important than sexual reproduction through larvae. A single large colony damaged by a hurricane might inadvertently seed a dozen new colonies from its broken branches, each one a genetic clone of the parent, each one carrying the same successful genetic blueprint that allowed the parent to grow large in the first place. Night falls on the reef. Not suddenly, the way night falls on land when the sun drops below a sharp horizon, but gradually, the way light fades in a forest at dusk. The blue above deepens. Shadows lengthen. Fish that were active during the day retreat to crevices and caves, and the reef transforms. Creatures that hid during daylight emerge. Moray eels slide out of their holes. Octopuses uncurl from rocky crevices, and throughout the coral forest, the sea fans begin to feed. The transformation is subtle, if you're not looking for it, but it's remarkable when you know what to watch. During the day, many sea fans keep their polyps retracted, pulled back into their protective calyces. The surface of the fan looks relatively smooth, almost waxy, but as darkness comes, as the risk from polyp-eating fish diminishes, the polyps emerge. Hundreds of them, thousands, each one extending its 8 delicate tentacles into the current, creating what looks like a coating of fine fur across the entire surface of the fan. The colony is feeding. Each polyp is a tiny mouth surrounded by 8 arms, and each arm is lined with specialized cells called nematocysts. These are the same stinging cells that make jellyfish dangerous and give anemones their subtle sting. But in sea fans, they're much gentler, designed not to deter large predators, but to capture microscopic prey. The current brings the food. This is the genius of the sea fan's strategy. It doesn't have to hunt. It doesn't have to move. It simply stands in the path of the ocean's endless conveyor belt and catches what flows by. Zooplankton, primarily, tiny copepods, larval crustaceans, the eggs of fish and invertebrates, organisms so small that they drift with the currents rather than swimming against them. These are the foundation of the ocean's food web, the tiny creatures that feed on even tinier phytoplankton, converting the energy of sunlight captured by algae into animal protein that everything else in the ocean depends on in one way or another. When a piece of zooplankton bumps into an extended tentacle, the nematocysts fire. It happens too fast to see. Even if you were watching with the finest microscope, the specialized cells discharge their coiled threads, some sticky, some barbed, all designed to entangle or penetrate the tiny prey. The tentacle then curls, bringing the captured food toward the polyp's mouth at the center of the 8 tentacles. The mouth opens. The food is pushed inside into the gastrovascular cavity, the simple digestive chamber that occupies the interior of each polyp. Here, enzymes begin to break down the prey, but the nutrition doesn't stay local. Remember those canals that connect all the polyps throughout the colony. Once food is partially digested, nutrients flow through these channels, distributing the energy from this one captured copepod to polyps throughout the colony. Some of those polyps might have caught nothing. Some might be in parts of the fan where current flow is less optimal, but they all share in the bounty. The colony pools its resources. But capturing zooplankton isn't the only way sea fans feed. Many species, particularly those in shallow, well-lit waters, have formed that remarkable partnership with zooxanthellae, those symbiotic algae living inside their tissue. During the day, when the sun shines down through the water, these algae are busy with photosynthesis. They're using light energy to convert carbon dioxide and water into sugars and oxygen. The sugars, or more precisely the various organic compounds the algae produce, leak from the algae cells into the surrounding coral tissue. The polyps absorb these compounds and use them for energy, for growth, for reproduction. It's been estimated that in some shallow water gorgonians, the zooxanthellae might provide more than half of the colony's nutritional needs. Some species might get 70 or even 80% of their energy from these internal farmers. This partnership is mutually beneficial, a true symbiosis. The coral provides the algae with a safe place to live, protected from grazers that would eat free-floating phytoplankton. It provides nutrients, nitrogen and phosphorus from the coral's waste products, which the algae need but which are often scarce in tropical waters. And it provides access to sunlight, which is everything to a photosynthetic organism. In return, the coral gets a steady supply of food without having to catch it, a baseline of nutrition that sustains it even when plankton is sparse. This is why you often see the most luxuriant gorgonian growth in the well-lit shallows of tropical reefs, where both feeding opportunities and photosynthesis can operate at their peak. But not all sea fans have this partnership. Deep water species, living in depths where sunlight fades to nothing, cannot host photosynthetic symbionts. These species rely entirely on capturing food from the water. They're suspension feeders, pure and simple, and their survival depends entirely on the currents bringing them enough plankton to sustain their slow metabolism and their glacial growth. In the deep sea, where food is generally scarce compared to shallow waters, this means that Gorgonian forests tend to grow in specific locations where currents are strong and where upwelling or other oceanographic features concentrate plankton. Cedar mounts, underwater mountains that rise from the abyssal plain, often host spectacular Gorgonian forests because currents flowing up and over these mountains create turbulence that concentrates plankton. The feeding rates, the amount of food a sea fan colony can capture in a day or a week or a year, depend on many factors. Current speed is one of the most important. Too slow and not enough water flows through the fan to deliver sufficient food. Too fast, and the polyps might not be able to extend their tentacles effectively, or the forceful current might actually reduce feeding efficiency. There's an optimal range, and it varies by species. Plankton concentration matters too, obviously. A small colony in a rich current might capture more food than a large colony in plankton-poor water, and the size and structure of the colony itself plays a role. Larger colonies with more polyps can potentially capture more total food, but there can be diminishing returns. Polyps downstream from other polyps are feeding on water that's already been partially filtered. The interior of a very dense fan might get less effective water flow than the outer edges. Sea fans have evolved their architecture to optimize this complex equation. The spacing of branches, the orientation of the fan perpendicular to the current, the tendency of branches to develop in ways that maximize the surface area exposed to flowing water. All of these are solutions to the problem of feeding while anchored in place, and the colonies continuously adjust. Young sea fans don't always orient correctly at first. They might grow in random directions initially, but as they mature, as they grow large enough that they extend up out of the boundary layer near the bottom and into the main current flow, they begin to reorient. The holdfast can twist slightly in response to mechanical stress from the current. Growth becomes preferentially directed in the plane perpendicular to flow. Over years, the colony aligns itself, positioning its fan to catch the maximum amount of water, to filter the maximum amount of food, to grow as large as the environment will support. There's something almost magical about the moment when a coral spawns. It happens once a year for most species, sometimes more often, sometimes less. The exact timing varies, triggered by a complex interplay of environmental cues that scientists are still working to fully understand. Water temperature is often involved, day length, the phase of the moon, perhaps even more subtle signals, chemical cues in the water or patterns in the current that tell the coral when conditions are right. But when the time comes, when all the signals align, the transformation is spectacular. In species that release their gametes into the water, the spawning can be a dramatic event. Imagine diving on a reef at night, your dive light cutting through the darkness, and suddenly the water is full of tiny pink or orange or white particles. Millions of them, billions of them, rising from the sea fans and other corals like an underwater snowstorm in reverse. These are gamete bundles, packets of eggs and sperm released simultaneously by thousands of coral colonies across the reef. Each sea fan colony, if it's mature enough, if conditions are right, releases its genetic material into the current. The bundles float upward toward the surface, buoyant with lipids, and as they rise they break apart. Eggs and sperm separate and drift, sometimes for hours, mixing and mingling in the water column. Fertilization is a numbers game. Each egg needs to encounter a sperm from a different colony to create a genetically diverse offspring. But the ocean is vast and the gametes are tiny. Many eggs will never be fertilized. Many will be eaten by fish or filter feeders, but enough succeed. When egg and sperm meet, when genetic material combines, the fertilized egg begins to develop. Cell division starts almost immediately. One cell becomes 2, becomes 4, becomes 8. Within hours or days, depending on the species and the water temperature, the developing embryo transforms into a planula larva. This planula is a remarkable little creature. It's barely visible to the naked eye, perhaps a millimeter long, shaped like a tiny elongated football, and covered with cilia, fine hairs that beat in coordinated waves to propel it through the water. The planula doesn't feed. It lives off the yolk provided by the egg, the stored energy that will sustain it for days or weeks while it searches for a place to settle. and it swims, though perhaps swim is too strong a word. It drifts mostly, carried by currents, but it can also make small vertical movements, rising toward light or sinking toward darkness, perhaps helping to keep itself in favorable current flows that might carry it to suitable habitat. The larval period varies considerably among species. Some sea fan larvae are competent to settle within days of fertilization, ready to attach and metamorphose after only a brief time in the plankton. Others might drift for weeks or even months, traveling vast distances on ocean currents. These long larval periods can be advantageous, allowing the species to disperse widely, to colonize new reefs far from the parent colonies. But there are costs. The longer a larva drifts, the more likely it is to be eaten, to starve, or to be carried into unsuitable habitat where it cannot survive. It's a trade-off between dispersal and mortality, and different species have evolved different strategies. When a planula finally encounters suitable substrate, the settlement process begins. The larva has chemical sensors on its surface, ways of detecting the qualities of the surface it's touching. It's looking for several things: a hard surface. First of all, sea fans can't settle on soft sand or mud. They need rock or coral skeleton, or some other firm anchor point, but not just any rock will do. The larva can detect chemical cues that indicate whether this is a healthy reef, whether other corals are successfully growing nearby. It might prefer surfaces covered with crustose coralline algae, those pink or purple encrusting algae that often indicate good conditions for coral growth. It might avoid surfaces covered with fine sediment or with certain species of algae that inhibit coral settlement. Once the larva decides to settle, metamorphosis begins. This is a one-way transformation. The ciliated planula attaches to the rock and begins to change. The cilia are reabsorbed. The body flattens and spreads, forming the basal disc that will anchor it permanently to this spot. The mouth forms, and around it, the first tentacles begin to develop. 8 of them, the signature feature of all octocorals. The larva, which has been surviving on stored energy until now, becomes a polyp, able to feed and grow. And crucially, it begins to secrete skeleton, laying down the first bits of gorgonin or calcite that will eventually form the internal axis of a large colony. But reproduction isn't always sexual. Many sea fans can also reproduce asexually through fragmentation. A storm might break a branch from a colony. That branch, if it settles on suitable substrate, can potentially reattach and grow into a new colony, a perfect genetic clone of its parent. In some environments, this might be the dominant form of reproduction. Researchers studying gorgonian populations have sometimes found genetic evidence of extensive cloning. Many colonies in an area all genetically identical, all likely descended from fragments of a single successful parent colony. There's also the possibility of budding new colonies from existing ones, though this is less well documented in sea fans than in some other coral groups. And some species might produce specialized asexual propagules, small structures that detach from the parent and can drift a short distance before settling to found a new colony. The balance between sexual and asexual reproduction varies by species and by environment. Sexual reproduction produces genetic diversity, which gives the population the variability it needs to adapt to changing conditions. Asexual reproduction is energetically cheaper and can rapidly generate new colonies in an area where conditions are known to be favorable. In a stable environment, asexual reproduction might dominate, but when conditions change, when disease sweeps through, or when water temperature shifts beyond the normal range, the genetic diversity created by sexual reproduction becomes crucial for survival. If you could shrink yourself down small enough to wander through a sea fan colony the way you might walk through a forest, you'd find yourself in a city, a bustling metropolis of organisms, most of them too small to see with the naked eye, but collectively creating an ecosystem of staggering complexity. The sea fan itself is the architecture, the structure that makes all of this possible. Its branches are the boulevards and alleyways. Its polyps are the buildings, and living among them, using them, depending on them, are hundreds of other species. Start with the obvious residents. There are brittle stars, those delicate relatives of sea stars, clinging to the branches with their long, flexible arms. They climb up into the fan to reach higher into the water column, where current is stronger and food more abundant. From their perch, they extend their arms into the flow just like the polyps, catching passing plankton with tube feet lined with sticky mucus. Some species of brittle stars seem to live almost exclusively on sea fans, their entire life cycle playing out in the branches of these corals. You might find a dozen brittle stars on a single large fan, sometimes more, each one carefully positioned to maximize its feeding efficiency without interfering too much with its neighbors. The brittle stars demonstrate one of the key ecological functions sea fans provide: vertical structuring of the habitat. On a flat rocky bottom, every filter feeder is at the same height, competing for the same slice of water column. But when a sea fan grows upward, it creates different heights, different zones where different organisms can position themselves. A brittle star at the tip of the tallest branch is feeding from faster, cleaner water than one at the base. Smaller organisms can shelter deeper in the colony while larger ones occupy the outer branches. This vertical layering increases the total number of individuals the habitat can support, what ecologists call carrying capacity. Basket stars take this to an extreme. These relatives of brittle stars have arms that branch repeatedly, creating intricate tangles that can span half a meter or more. During the day, they curl up into tight balls, hiding their delicate arms from potential predators. But at night, they unfurl, climbing to the tips of sea fan branches or other high points on the reef, and they spread their branching arms into the current like living nets. They're suspension feeders too, catching even smaller plankton than the brittle stars, filtering the water with thousands of tiny arm branches. A basket star perched on a sea fan creates a striking image: two colonial filter feeders using the same strategy in the same location, one a coral with hundreds of polyps, the other a single animal with hundreds of branching arms. The relationship between basket stars and sea fans illustrates an important ecological principle: niche partitioning. They're both suspension feeders in the same location, potentially competing for the same food, but they don't exclude each other because they specialize slightly differently. Basket stars catch smaller particles than sea fan polyps typically capture. They extend higher into the water column. They feed at night when many fish are less active, reducing predation risk. By dividing the available resources in these subtle ways, both can coexist, both can thrive, increasing the diversity and complexity of the reef community. Smaller still are the copepods, microscopic crustaceans that hide among the sea fan's branches during the day and emerge at night to graze on the film of algae and bacteria that coats every surface in the ocean. Some copepod species are parasites, specialized to feed on sea fans, using modified mouthparts to pierce the coral tissue and suck fluids. These parasites can sometimes form galls, swellings in the coral tissue where the copepod lives, protected from the outside world. The galls are created by the coral's response to the parasite's presence, tissue growing abnormally around the invader, inadvertently providing it with a safe chamber. Other copepods are commensals, living in the protection of the fan but not harming it directly, feeding on material in the water or grazing on surface films but not attacking the coral itself. And some might even be beneficial, grazing on algae that might otherwise overgrow the fan or removing sediment that could smother polyps. The exact nature of many of these relationships remains unclear. It's difficult to determine whether a tiny copepod is helping or hurting its host, whether its effects are significant or negligible. But collectively, these microscopic organisms add to the complexity of the community, to the web of interactions that makes the sea fan more than just a coral but rather a complete ecosystem. There are tiny shrimp, translucent as glass, living their entire lives in the branches of a single sea fan. Some species of shrimp are so specialized to this lifestyle that they've evolved coloration that matches their host coral. A shrimp living on a purple sea fan might be purple itself, perfectly camouflaged against the coral tissue. These shrimp defend their territory aggressively, driving off other shrimp that try to colonize the same fan. A large sea fan might support a mated pair of shrimp and their offspring, a little dynasty spanning generations. The shrimp aren't just passive residents. Some species actively clean their host coral, picking off bits of debris, perhaps even consuming small parasites or diseased tissue. This creates a mutualistic relationship where both partners benefit. The shrimp gets shelter and perhaps some nutrition from the coral, while the coral gets cleaning services that might improve its health and reduce infection risk. Other shrimp species are less helpful, feeding on the coral tissue itself, essentially farming their host by grazing lightly without killing it, but certainly not providing any benefit in return. Fish use sea fans too, though in different ways. Some species, particularly juveniles of larger reef fish, hide among the branches, using the coral as shelter from predators. The gaps in the lattice are perfect for small fish, large enough to swim through freely, but complex enough to make it hard for a larger predator to maneuver. The three-dimensional structure creates what ecologists call structural complexity, providing hiding places, ambush points, and territories that simply wouldn't exist on a flat, featureless bottom. Groupers might hunt among sea fans, relying on their excellent camouflage and patient stalking to catch smaller fish sheltering in the coral branches. Their hunting strategy depends on the structure the coral provides, places where they can position themselves in ambush, spaces where fleeing prey have limited escape routes. Seahorses and their relatives, the pipefish, sometimes cling to sea fan branches with their prehensile tails, their bodies swaying with the coral as both move together in the current. They're masters of camouflage themselves, with body shapes and coloration that often mimic the branching patterns and colors of the corals they inhabit. The most famous fish association is probably the pygmy seahorses. These remarkable little creatures are among the smallest seahorses in the world, rarely more than 2 centimeters long. They live exclusively on certain species of sea fans in the Indo-Pacific, and they're so well camouflaged that they were discovered by accident when a scientist collected a sea fan for study and only later noticed the tiny seahorses clinging to it. The seahorses match not only the color of their host coral, but even the texture, developing bumps and protrusions on their skin that mimic the polyps of the sea fan. They spend their entire adult lives on a single coral colony, feeding on tiny crustaceans that drift past, reproducing in the safety of the branches, never venturing away from their living home. But not all residents are benign. There are predators that specialize in eating sea fans. The flamingo-tongued snail, with its beautiful leopard-spotted mantle, crawls across sea fans grazing on the living tissue. It doesn't kill the coral outright, but it leaves bare patches where the tissue has been consumed, exposing the skeleton beneath. A heavy infestation of these snails can severely damage or even kill a sea fan. Similarly, certain species of nudibranchs, those colorful sea slugs, feed exclusively on gorgonians. They too crawl across the surface, rasping away tissue with their radula, a ribbon of teeth, unlike anything else in the animal kingdom. Some nudibranchs go a step further, sequestering the defensive chemicals from the sea fan's tissue and using them for their own protection, recycling the coral's weapons for themselves. And there are worms, polychaetes, that burrow into the skeleton itself, creating galleries and tunnels that can weaken the structural integrity of the coral. Some of these worms might actually help the coral in a strange way by creating water flow through their tunnels that helps distribute nutrients, though if they become too numerous, they can cause branches to break. There are also gall-forming parasites, including some copepods that burrow into the tissue and create swellings around themselves, little tumors of coral tissue that house the parasite and protect it from the outside environment. Even algae and other corals will sometimes try to colonize a sea fan. When a portion of a fan dies, when tissue is lost due to disease or predation, the exposed skeleton becomes real estate. Algae might settle on it. Other corals, particularly encrusting forms, might overgrow the dead portions and even begin encroaching on living tissue. Some hydrozoans, relatives of corals that form branching colonies or encrusting mats, compete with sea fans for space. If they overgrow the living tissue, if they manage to cover the polyps and prevent them from feeding, they can kill sections of the fan or even entire colonies. The sea fan doesn't take this passively. It has defenses. Many species produce toxic or distasteful compounds, secondary metabolites, that deter at least some predators and competitors. These chemical defenses vary widely among species. Some are diterpenes, organic compounds that can have antimicrobial, antifungal, or anti-fitting properties. Researchers have even observed bottlenose dolphins deliberately rubbing against certain sea fans, seemingly taking advantage of these compounds' antimicrobial effects to treat skin infections. The sclerites embedded in the tissue provide physical defense too, making the flesh gritty and unpleasant to eat. But perhaps the most important ecological role sea fans play is simply being structure. In many reef environments, particularly in deeper areas or on rubble slopes where stony corals are less abundant, sea fans and other gorgonians are the dominant three-dimensional structure. They turn a flat, relatively featureless bottom into a complex vertical habitat. They create the architecture that allows other species to exist in places they otherwise couldn't. Without the sea fans, the brittle stars would have nowhere to climb. The pygmy seahorses would have no home. The fish would have no shelter. Remove the sea fans and the entire community collapses. There are thousands of species of sea fans scattered across the world's oceans from the tropics to the poles. From shallow reefs you can snorkel to abyssal depths that require submarines to explore. Each species has evolved its own particular shape, its own coloration, its own set of adaptations to the specific environment where it lives. The diversity is staggering, though most people encounter only a handful of species, unless they're scientists or dedicated enthusiasts willing to study the minute differences that distinguish one gorgonian from another. In the Caribbean and Western Atlantic, the purple sea fan, Gorgonia ventellina, is perhaps the most familiar. This is the species that appears in countless underwater photographs, the one with the characteristic fan shape and the striking purple or lavender coloration on its main branches. It grows in shallow to moderate depths, typically from a few meters down to perhaps 15 or 20 meters, where there's enough light for its symbiotic zooxanthellae and enough current to bring plankton. Large colonies can reach well over a meter in height and width, spreading their lattice of branches perpendicular to the prevailing surge. The purple color is most intense in the main stems and branches, while smaller branchlets might be paler, sometimes yellowish or even white where new growth is most active. A close relative, the Venus sea fan, Gorgonia flebellum, looks similar at first glance, but has subtle differences. Its branches tend to be more three-dimensional, less compressed in the plane of the fan. It produces small accessory fans that grow out sideways from the main fan, creating a more complex structure. And it lives in slightly different habitat, often in areas with less wave action than the purple sea fan prefers. Then there are the sea whips, Gorgonians that have abandoned the fan shape entirely. These grow as single tall branches, or sometimes with a few minimal side branches, like undersea saplings reaching toward the surface. They can be brightly colored, reds and yellows and oranges, and they grow in sandy areas between reefs or on reef slopes where the fan shape might not be advantageous. Instead of growing perpendicular to the current, sea whips grow vertically, and their polyps can feed from water moving in any horizontal direction. Sea rods are stouter, more robust, with thicker branches that can handle higher energy environments. They too might branch only minimally, or they might create a more bush-like structure rather than a single plane. Sea plumes develop delicate feathery branches extending from a central axis, looking indeed like underwater feathers or plumes swaying in the current. In the Mediterranean and Eastern Atlantic, you find different species adapted to cooler waters. The pink sea fan, Eunicella verrucosa, grows in British waters and along the coasts of France and Spain. Unlike its tropical cousins, this species lives without symbiotic algae, relying entirely on captured plankton for nutrition. It grows extremely slowly, as we discussed earlier, perhaps a centimeter a year, and it can be pink, white, or orange. Because of its slow growth and limited range, it's considered vulnerable, protected by conservation laws in several countries. The Indo-Pacific hosts a tremendous diversity of gorgonians, including some of the most bizarrely shaped and brightly colored species anywhere. There are species with branches that twist like corkscrews, species with such tight branching that they look like colorful underwater broccoli, species that form encrusting mats rather than upright fans. The diversity reflects the tremendous age and stability of Indo-Pacific reefs, which have allowed more time for speciation, for the evolution of specialized forms. And then there are the deep-sea species, the gorgonians that live in eternal darkness on the slopes of seamounts and the edges of continental shelves. Some of these create massive tree-like colonies. Paragorgia arborea, the bubblegum coral, can grow to 3 meters tall or more, with thick branches creating complex three-dimensional structures that host hundreds of associated species. It's called bubblegum coral because of its bright pink or red color and the slightly bumpy texture of its branches. These massive colonies are incredibly slow-growing and can be centuries old. Primnora resediformis, another deep-water species, forms similarly large colonies and is particularly common in the North Atlantic and North Pacific. It creates dense thickets on hard substrate, turning stretches of seafloor into genuine coral forests. Fishermen call these coral trees, and the term isn't far off. Walking among them in a submersible is said to be uncannily like flying slowly through a forest, with towering structures on all sides creating a canopy overhead and smaller colonies forming an understory below. Bamboo corals, named for the segmented appearance of their skeletons, are another deep-sea group. Their axes alternate between calcified sections and flexible gorgonin nodes, creating a structure that's jointed like bamboo. This might allow them to flex in deep currents without breaking, or it might be related to their growth pattern. But the exact evolutionary advantage isn't entirely clear. What is clear is that they can be spectacularly beautiful, with some species displaying bright yellows and oranges against the dark deep-sea background. Gold corals, precious corals valued in jewelry making, are gorgonians found in deep water around volcanic islands, particularly in the Pacific. They're among the longest-lived marine organisms known, with some specimens radiocarbon dated to over 2,000 years old. Their skeletons, when polished, have a beautiful golden sheen, which has made them targets for harvesting. Fortunately, their deep habitat and slow growth have led to protective measures in some areas, recognizing that these ancient colonies cannot sustain harvest pressure. Black corals, though not technically gorgonians, they belong to a different order, Antipathuria, are often grouped with them ecologically. They grow in similar environments, create similar structures, and serve similar ecological roles. Their skeletons are made of a black proteinaceous material that can be carved and polished, making them valuable in jewelry. Like the gorgonians, they're slow-growing and long-lived, with some specimens estimated to be thousands of years old. The colors we see in all these species come from various sources. Symbiotic algae contribute browns and greens. Carotenoid pigments produce yellows, oranges, and reds. Other organic compounds create purples and blues. Some species can change color slightly in response to light levels or other environmental factors, and the color can vary within a species across its range. A purple sea fan in Bermuda might have slightly different coloration than one in the southern Caribbean, perhaps due to different water chemistry or different populations of symbiotic algae. Not everything in the coral forest is timeless and peaceful. There are threats. Some natural, part of the endless cycle of growth and destruction that has shaped reef ecosystems for millions of years. Others new or at least intensified beyond natural levels by human activity. And sea fans, with their slow growth and their sessile nature, with their reliance on specific environmental conditions, are vulnerable to many of these threats. Disease is perhaps one of the most visible. In the Caribbean particularly, sea fans have suffered from outbreaks of aspergillosis, a fungal infection caused by Aspergillus sidawi. This fungus, normally found in terrestrial environments, somehow made its way into the ocean and found gorgonians susceptible to infection. It enters through wounds or areas of damaged tissue and spreads through the coral, causing lesions, creating purplish patches of damaged tissue and sometimes killing entire colonies. Large-scale epizootics, disease outbreaks affecting many colonies across wide areas, have been documented multiple times in the Caribbean since the fungus was first identified in sea fans in the late 1990s. The susceptibility to aspergillosis seems to increase when sea fans are stressed by other factors. High water temperatures, pollution, low salinity from heavy rainfall or river discharge. All of these can weaken the coral's immune responses and make infection more likely. Researchers studying the disease have found that it doesn't kill all sea fans, at least not immediately. Some colonies seem more resistant than others, suggesting genetic variation in disease resistance within the population, and some affected colonies can recover, walling off infected tissue and regrowing around the damaged areas, though this recovery process can take years. There are other diseases too, other pathogens waiting to exploit any weakness. Some are caused by bacteria, others might be viral, though coral viruses are still poorly understood. There are also parasitic protists, single-celled organisms that can infect coral tissue. Each pathogen has its own ecology, its own patterns of spread, its own effects on the host, and in some cases, we don't even know what's causing the disease we observe, only that colonies are dying and we can't identify the causal agent. Climate change looms over everything. Rising ocean temperatures affect sea fans, both directly and indirectly. Directly because, like all organisms, corals have temperature ranges within which they function optimally. Push the temperature too far beyond that range, and metabolic processes start to fail. Polyps might retract and stop feeding. Growth slows or stops. Reproduction might fail. In species with symbiotic algae, high temperatures can trigger coral bleaching. The algae, stressed by heat and producing harmful oxygen radicals as a byproduct of disrupted photosynthesis, are expelled by the coral host. The colony loses its color, turning pale or white as the algae disappear, and more importantly, it loses a major source of nutrition. If temperatures return to normal quickly, the algae can recolonize and the coral recovers, but if the heat stress continues, the coral starves. Gorgonians generally seem somewhat less prone to bleaching than stony corals, perhaps because their symbiotic relationships are slightly different, or perhaps because many species don't rely as heavily on photosynthesis. But they're not immune. Bleaching events have been documented in sea fans, and it's unclear how severe climate change would have to become before it causes widespread mortality in these corals. Indirectly, climate change affects sea fans through its impacts on ocean chemistry. As atmospheric carbon dioxide levels rise, the ocean absorbs more CO2, which dissolves in seawater, forming carbonic acid. This ocean acidification doesn't make the ocean actually acidic, at least not yet, but it does shift the pH downward, making the water less alkaline than it has been for millions of years. This shift affects many marine organisms, particularly those that build calcium carbonate structures. Sea fans with calcified sclerites might find it harder to produce these structures. The gorgonin skeleton might not be directly affected, but changes in water chemistry can have subtle effects on many biological processes. We don't yet know how serious ocean acidification will be for gorgonolians, but it's an area of active research. Storm damage is a natural threat that climate change might be intensifying. Hurricanes and tropical storms have always battered reefs, breaking coral colonies, flipping boulders, scouring away sediment and sometimes entire sections of reef. Sea fans, with their relatively delicate structure, are particularly vulnerable to storm damage. A large hurricane can break branches from colonies or rip entire fans from their holdfasts. In shallow areas where wave action is most intense, few sea fans survive in place for long periods. They grow, they reach a certain size, and then a storm breaks them or tears them loose, and they start over. But broken fragments, as we discussed, can sometimes establish new colonies. So storm damage is both destructive and creative, removing large established colonies, but potentially spreading genetic material around the reef, creating new growing points from fragments. Over evolutionary time, this cycle of destruction and regeneration has been a constant part of the Gorgonian life strategy. Human impacts add new threats or intensify old ones. Physical damage from anchors, from boat groundings, from careless divers or snorkelers, is an ongoing problem in many popular reef areas. A dragged anchor can rip across a reef, leaving a scar of broken coral that might take decades to heal. A diver fin kicking into a sea fan can break branches that took years to grow. Most of this damage is accidental, the result of carelessness or ignorance rather than malice, but the effects are the same. Fishing, particularly bottom trawling, is devastating to deep-sea Gorgonian forests. A trawl net dragged across the seafloor doesn't discriminate between target fish and bycatch, between fish and invertebrates, between free-swimming and sessile organisms. It crushes or uproots everything in its path. Coral forests that took centuries or millennia to grow can be reduced to rubble in minutes. Researchers' submersible surveys have documented the before and after impacts of trawling in areas with deep-sea corals, and the transformation is heartbreaking. Where there were once magnificent 3-meter-tall Primnoa colonies creating complex habitat, afterward there's bare rock strewn with broken coral fragments. Some countries have responded by creating protected areas, zones where bottom trawling is prohibited to preserve deep-sea coral habitat. The logic is both ecological and economic. These coral forests support commercially important fish populations. Protecting the corals protects the fish, which ultimately benefits the fishery. It's a long-term conservation strategy rather than short-term exploitation. Pollution is another human impact. Sediment from coastal development, nutrients from agricultural runoff and sewage, chemicals from industrial activities. All of these make their way into coastal waters and onto reefs. Heavy sediment loads can smother sea fans, coating their polyps and preventing them from feeding. Excess nutrients can fuel algae growth that competes with corals for space or that blocks light needed by symbiotic zooxanthellae. Some chemical pollutants might directly poison coral tissue or interfere with reproduction or larval settlement. And finally, there's collection for the aquarium trade and for curios. Sea fans dried and mounted make attractive decorations, and there's a market for them in some tourist areas. Live sea fans can be kept in specialized aquarium setups, though they're difficult to maintain and their slow growth means wild collected specimens take decades to replace. Most collection is probably small scale and localized, but in some heavily visited areas it could be significant. Several countries now protect sea fans specifically, prohibiting collection without permits. Far beneath the sunlit reefs where divers swim, far below the depth where photosynthesis is possible, far into the cold darkness where pressure mounts with every meter of descent, there are still sea fans. Different species, adapted to a very different world, but still recognizable as gorgonians, still building their colonies through the patient addition of polyp after polyp, still filtering the water for food, still creating structure that other organisms depend on. The deep sea is not the barren wasteland it was once thought to be. In recent decades, as submersibles have allowed scientists to explore deeper and deeper, we've discovered entire ecosystems in the abyss. Hydrothermal vent communities, powered not by sunlight but by chemical energy from the Earth's interior. Cold seep communities where methane and other chemicals leak from the seafloor supporting specialized bacteria and the animals that feed on them. And coral forests, Gorgonian gardens growing in places that see no sunlight at all, that might experience temperatures just a few degrees above freezing, that exist under pressure that would crush an unprotected human instantly. These deep-sea gorgonians have evolved remarkable adaptations to their environment. They don't have symbiotic algae, obviously. There's no light for photosynthesis. They rely entirely on suspension feeding, on capturing what little plankton drifts down from the productive surface waters far above. And in the deep sea, food is scarce. The rain of organic material from above, the marine snow of dead plankton and fecal pellets and other detritus is thin. Deep-sea organisms generally have slower metabolisms than their shallow water relatives, and they grow much more slowly. The pressure itself presents challenges that deep-sea gorgonians have solved in ways we're still working to understand. At depths of thousands of meters, the pressure can exceed several hundred atmospheres. This pressure affects the structure of proteins, the fluidity of cell membranes, the solubility of gases. Deep-sea organisms have specialized biochemistry that functions normally under these extreme pressures. Their proteins are structured differently than those of shallow water species, maintaining proper folding and function despite the crushing force. Their cell membranes have different lipid compositions, remaining flexible and functional rather than becoming too rigid. The cold is another challenge. Temperature affects every biological process, slowing molecular movement, reducing reaction rates, affecting enzyme efficiency. Deep-sea gorgonians have evolved enzymes that work efficiently at low temperatures, remaining active where shallow water enzymes would barely function. Their metabolism runs slower than their warm water cousins, but it runs reliably, maintaining the coral through decades and centuries of growth in the perpetual chill of the abyss. Food limitation has driven the evolution of extreme feeding efficiency. Deep-sea gorgonians have polyps that can extend remarkably long tentacles relative to their body size, creating larger capture surfaces. Some species have specialized nematocysts that are more effective at capturing the types of particles common in deep water. They might be able to feed on smaller particles than shallow water species, extracting nutrition from detritus that other filter feeders would pass through their feeding structures. We've already mentioned the extreme ages. Some of these corals can reach 2,000 years, 4,000 years, possibly even older. But it's worth considering what this means. These are individual organisms, continuously alive, continuously growing, for longer than recorded human history. When we damage or kill a deep-sea gorgonian colony, we're not just removing a coral, we're erasing thousands of years of slow growth, thousands of years of accumulated ecological relationships, thousands of years of history. The deep-sea species tend to be different in form from shallow water sea fans. Many are tree-like, rather than fan-shaped. This might be because current patterns are different in the deep sea, less unidirectional, more variable. A fan shape only works if currents predominantly flow from one direction. If currents swirl and change, a three-dimensional bush or tree form might be more effective. Or it might be that without light as a limiting factor, there's no advantage to growing in a single plane to maximize light exposure, so the constraint that produces the fan shape is removed. The colors are often still vivid, despite the darkness. bright reds, pinks, oranges, whites. These colors aren't for the benefit of any observer, since no light exists to reveal them in their natural environment. They're likely byproducts of the chemical compounds the corals produce, compounds that might have defensive functions or metabolic roles that happen to be pigmented. When a submersible's lights illuminate a deep-sea Gorgonian forest for the first time, the burst of color against the black background is startling. These deep forests grow on specific substrate, hard rock, usually. In the deep sea, most of the bottom is soft sediment, mud accumulated over millions of years, and Gorgonians can't attach to that. So they're found where geological features create hard surfaces. Sedamounts, underwater volcanoes that rise from the abyssal plain, are often covered with Gorgonian forests because they're made of hard volcanic rock. The slopes of continental margins, where rocky outcrops interrupt the sediment layers, host corals. Ridge systems, where tectonic plates diverge and new oceanic crust forms, can have corals. And occasionally, hard substrate appears in unexpected places. Rocks dropped by melting icebergs, whale bones providing a temporary hard surface for larvae to settle on. Current patterns at depth determine where the largest forests grow. Gorgonians need currents to bring food, but currents in the deep sea are often sluggish. Exceptions occur where oceanographic features concentrate flow. Water flowing up and over a seamount creates turbulence and upwelling that brings nutrients from below and debris from above. Bottom currents channeled through submarine canyons can create local areas of stronger flow. These are the places where deep-sea corals thrive, creating forests that rival any shallow reef in complexity, if not in total biomass. The ecosystems that depend on these deep-sea coral forests are only beginning to be understood. We know that fish aggregate around them. Commercially important species like rockfish, orange ruffy, and various deep-sea snappers are often found in association with coral forests. Whether they use the corals for shelter, for feeding grounds where smaller prey congregate, or for spawning habitat isn't always clear, but the association is strong enough that fishermen have long known to trawl in areas with corals for better catches. This is what led to the widespread destruction of these habitats, ironically from the very industry that benefits from their existence. Other residents include the same sorts of suspension feeders we see in shallow water: brittle stars, crinoids, sea urchins, various worms and crustaceans. There are deep-sea squat lobsters that look like tiny lobsters with their tails curled beneath them, perched on coral branches. There are sponges growing among the corals, their own filter feeding strategies complementing those of the Gorgonians. There might be sea stars, sea cucumbers, tube worms. The diversity is high compared to nearby areas without corals, demonstrating again the fundamental ecological role that Gorgonians play as habitat creators. Some deep-sea Gorgonians host symbiotic relationships we're only beginning to discover. There are parasitic copepods that create galls, swellings in the coral tissue where they live protected from the environment. There might be specialized polychaete worms or other organisms that live only in association with particular coral species. Each new submersible dive, each new sample collected and examined carefully, reveals organisms never seen before, adding to our understanding of these remote ecosystems. For most of human history, sea fans were curiosities, pretty things to collect on beaches, to dry and mount as decorations. Sailors brought them back from tropical voyages as exotic souvenirs. Naturalists in the 18th and 19th centuries collected and described them, creating the first taxonomic catalogues, grouping species into genera and families based on their gross morphology. But understanding how they actually lived, how they grew, how they reproduced, how they fit into the ecology of reefs, that required tools and techniques that didn't exist until relatively recently. The development of scuba diving in the mid-20th century revolutionized marine biology. For the first time, scientists could spend extended periods underwater, observing organisms in their natural habitat, rather than just examining dead specimens hauled up in nets. Early coral researchers began to document the basic biology of sea fans. They described feeding behavior, watching polyps extend their tentacles at night. They marked individual colonies and measured their growth over years, establishing the first reliable growth rate estimates. They collected samples and examined them under microscopes, discovering the symbiotic zooxanthellae, the microscopic sclerites, the complex internal anatomy. Submersibles opened up the deep sea. Manned submersibles like Alvin allowed scientists to descend to depths of thousands of meters, to observe deep-sea coral forests firsthand and collect samples carefully rather than blindly dredging them up and destroying them in the process. Remotely operated vehicles, ROVs, extended the reach even further, allowing exploration of the deepest parts of the ocean without risking human lives. These vehicles carry cameras, lights, manipulator arms for collecting samples, and sensors for measuring water properties. The footage they've collected has transformed our understanding of the deep ocean. Modern molecular techniques have revolutionized taxonomy and ecology. By sequencing DNA from coral tissue, scientists can establish evolutionary relationships between species, creating phylogenetic trees that show how different gorgonians are related to each other and when different lineages diverged. This has revealed that many species we thought were closely related based on their appearance are actually quite distant, while some species that look very different are close relatives. Form, it turns out, can be misleading. Molecular data provides a more reliable guide to actual evolutionary history. Genetic studies have also revealed patterns of reproduction and larval dispersal. By sampling colonies across a species range and looking at genetic variation, researchers can infer how much gene flow occurs between populations. High genetic similarity between distant populations suggests either recent connection via larval dispersal or very little genetic drift, implying large, stable populations. Lots of genetic structure, with populations in different areas being genetically distinct, suggests limited dispersal and long-term isolation. These patterns help us understand how species respond to fragmentation of their habitat, how quickly they might recover from local extinctions, how vulnerable they are to environmental changes. Radiocarbon dating has allowed us to determine the ages of deep-sea corals with remarkable precision. The technique works by measuring the ratio of carbon-14 to stable carbon isotopes in the coral skeleton. Carbon-14 is a radioactive isotope produced in the upper atmosphere by cosmic rays. It's incorporated into marine food webs along with stable carbon, and when a coral lays down new skeleton, it incorporates the carbon-14 from its food. Over time, the carbon-14 decays with a known half-life, so by measuring how much remains, you can calculate how long ago that skeleton was formed. This is how we know that some gorgonians are thousands of years old, how we've confirmed the incredible longevity of these slow-growing organisms. Chemical analysis of coral skeletons is revealing other information. The chemical composition of the skeleton can vary slightly depending on water temperature, chemistry, and other factors when it was laid down. By analyzing the chemistry along the length of a coral's axis, researchers can potentially reconstruct past ocean conditions, creating a timeline of environmental change spanning decades or centuries. This makes deep-sea gorgonians potential archives of ocean history, like tree rings recording the climate of past years but preserved in calcium and protein instead of wood. Research into coral reproduction has expanded dramatically in recent decades. Scientists have successfully spawned some sea fan species in captivity, watching the entire process from gamete release through larval development to settlement and metamorphosis. This work is crucial for conservation, potentially allowing restoration of damaged reefs by growing new corals in laboratories and outplanting them. It also answers basic biological questions about larval behavior, settlement cues, early growth, and survival rates. Studies of the chemical defenses that gorgonians produce have revealed a treasure trove of novel compounds. Some of these chemicals have potential medical applications: anti-inflammatory compounds, antimicrobial agents, compounds that might be useful in cancer treatment. Pharmaceutical researchers are screening extracts from various sea fan species, looking for useful molecules, though they're careful to collect specimens sustainably, and many are working on ways to synthesize the most promising compounds rather than harvesting corals from the wild. Research continues on the diseases affecting sea fans, trying to understand what causes outbreaks, what makes some colonies resistant while others succumb, and whether there are ways to manage the disease or help corals resist infection. Some scientists are investigating whether probiotics, beneficial bacteria that help exclude or suppress pathogens, could be used to protect corals. Others are looking at breeding programs to select for disease-resistant strains. Climate change research focuses on understanding how sea fans will respond to warming waters, changing ocean chemistry, and shifting current patterns. Experiments expose corals to controlled temperature increases, measuring how growth, feeding, and survival change. Models attempt to predict which species and which populations will be most vulnerable, where refugia might exist where conditions remain suitable, even as surrounding areas become too warm or too acidic. And there's still so much we don't know. Most sea fan species have never been studied in detail. We don't know their growth rates, their lifespans, their reproductive biology, their population structure, their ecological roles. We don't know how many species exist in total. Estimates suggest over 5,000 species of octocorals worldwide, but new species are described regularly, and some areas, particularly the deep sea, remain largely unexplored. Every submersible dive brings back samples of species never seen before, expanding our catalogue of marine biodiversity. Imagine you could hover weightless above a section of reef, looking down at the coral forest spread below you like a map. From this perspective, you begin to see patterns. The sea fans aren't distributed randomly. They cluster in certain areas, often where current flow is optimal. They orient almost uniformly perpendicular to the prevailing current, hundreds of fans all aligned like wind vanes, all pointing the same direction. Large colonies occupy the best positions, places where substrate is most stable and current most reliable. Smaller colonies fill in around them, in gaps, on less optimal substrate, in areas with slightly weaker flow. There's a structure to this forest, layers like the canopy and understory of a terrestrial forest. The tallest colonies, some over a meter high, create a kind of canopy, their upper branches extending into the fastest current. Below them, smaller colonies and other reef organisms exist in a modified flow environment, the current slowed somewhat by the presence of the larger colonies above. This creates microhabitats, areas where conditions differ from the surrounding reef, where different species adapted to slightly different conditions can coexist. Current plays the same role in this forest that wind and light play in terrestrial forests. It determines where things grow, how they're shaped, how they interact. Fast current brings more food, but creates more mechanical stress. Slow current is easier to live in, but might not deliver enough nutrition. Turbulent current, swirling and variable, favors different growth forms than steady unidirectional flow. Gorgonians and other suspension feeders position themselves to optimize their interaction with current, creating a landscape shaped by invisible forces. Seasonality exists even in tropical waters. The current patterns might shift with the seasons. Plankton abundance varies. Peaks tied to upwelling events or seasonal blooms of phytoplankton that cascade up the food web. Water temperature cycles through the year, warm in summer, cooler in winter, though the range might only be a few degrees. These cycles affect the coral forest. Growth might be faster in seasons with more plankton. Reproduction is often seasonal, timed to particular conditions. Some fish that use sea fans for shelter might be more abundant at certain times of year. Storm season brings dramatic change. In the Caribbean, hurricanes pass through between June and November, generating enormous waves and powerful currents. The first big storm of the season can transform a reef, breaking weakened colonies, scouring away sediment, rearranging the substrate. Afterward, the survivors remain, perhaps damaged but still alive, and the slow process of regrowth begins. At night, the reef comes alive differently. Polyps that stayed retracted during daylight extend their tentacles. Nocturnal fish emerge from their daytime hiding places. Invertebrates that hid from visual predators come out to feed. The sea fans spread their polyps into the darkness, filtering the water by touch and chemical sense rather than sight. Bioluminescence might flicker occasionally. A shrimp startled from its perch, a ctenophore drifting past trailing glowing tentacles. The transformation from day to night on a reef is not as dramatic as on land where the difference between daylight and darkness is absolute, but it's there, a change in the community, a shift in which species are active and which are dormant. Over longer timescales, the forest changes, colonies grow, some die, new larvae settle. Slowly over decades, the community composition shifts. What was a sparse scattering of young sea fans becomes a dense forest of mature colonies. Eventually, colonies begin to die, creating gaps that new larvae colonize. The forest becomes a patchwork of different ages, different stages of succession, a complex mosaic maintained by the balance between recruitment and mortality, between growth and destruction. This is especially apparent after major disturbances. A hurricane might wipe out large sections of a Gorgonian forest, leaving mostly broken stumps and scattered fragments. The recovery is slow. New larvae must settle. Fragments must reattach and grow. Young colonies must mature. It might take a decade before the forest begins to look dense again. 2 decades before it approaches its former complexity. In the deep sea, where growth is even slower, recovery from trawling or other damage might take a century or more. The sea fans themselves create the conditions for the forest's persistence. Their presence modifies the flow environment, creating areas of reduced current where other species can settle. Their 3-dimensional structure provides attachment points for organisms that couldn't survive on flat rock. When they die, their skeletons persist for a while, continuing to provide structure even after the living tissue is gone, only gradually being overgrown or broken down. They trap sediment, sometimes helping to stabilize the substrate, sometimes creating problems if too much sediment accumulates and smothers new recruits. And they create a legacy through their larvae. A large, healthy Gorgonian forest produces billions of larvae each year, though only a tiny fraction will survive to settlement. These larvae drift on currents, potentially traveling kilometers or tens of kilometers before settling. They might settle nearby, creating dense local populations of closely related individuals, or they might drift further, colonizing distant reefs, maintaining genetic connections between separated populations. The larvae are the forest's future, the next generation waiting to continue the slow work of growth and filtration that has characterized Gorgonians for millions of years. There's something humbling about standing on a reef, figuratively speaking, since we're weightless in the water, and looking at a sea fan that might be older than you are, that might have been growing before you were born, before your parents were born, possibly before your grandparents were born. Its existence spans generations of your family, and it's spent that entire time in this one spot, anchored to this one rock, filtering this water, feeding from this current. It hasn't moved, hasn't traveled. Its entire life, decades long, has been spent right here. And if it's a deep-sea species, if it's one of those ancient colonies from seamounts or continental slopes, the timescale becomes almost incomprehensible. 2,000 years. The colony was already centuries old when your ancestors, whoever they were, wherever they were living lives very different from yours. It was growing when historical events you learned about in school were current events. It was filtering plankton from deep Pacific water while empires rose and fell on distant continents, while languages evolved and changed, while humanity invented and refined technologies from agriculture to writing to metallurgy to industrialization to the digital age. And through it all, this coral just kept growing, slowly, patiently, polyp by polyp, millimeter by millimeter. This perspective, this deep time, it should change how we think about these organisms. They're not just decorations on the reef, they're not just pretty backgrounds for fish photography. They're living archives, tangible connections to the past, organisms that embody patience and persistence in a world that increasingly values speed and constant change. When we damage or kill a sea fan, especially an ancient deep-sea colony, we're not just removing one individual from the ecosystem, we're erasing history. We're ending a story that began when the world was different, when humans were different, when the ocean itself was different. The irony is that these slowest-growing, longest-lived organisms are often the most vulnerable to our fastest-paced activities. A trawling net pulled across a seamount for a few hours can destroy Gorgonian forests that took thousands of years to grow. An anchor dragged across a shallow reef for a few minutes can break sea fans that were growing before the boat's captain was born. The mismatch between the timescale of human activities and the timescale of Gorgonian recovery is profound. But there's also hope in this slowness. These organisms have persisted for millions of years through ice ages and warm periods, through changing ocean chemistry and shifting currents, through the rise and fall of other species and entire ecosystems. They've survived mass extinctions that killed off huge portions of marine life. They've adapted to changes we can barely imagine. This resilience suggests that with proper protection, with thoughtful management, sea fans can persist into the future as they've persisted through the past. Conservation efforts are increasing. More marine protected areas are being established, creating zones where destructive fishing practices are prohibited. Regulations are being developed to protect deep-sea coral forests from trawling. Research into coral restoration is advancing, with successful examples of sea fans being propagated and outplanted to damaged reefs. Education programs are teaching divers and snorkelers how to interact with reefs without causing damage. International treaties are being strengthened to regulate trade in corals and other reef organisms. But ultimately, the survival of sea fans, like the survival of so many marine species, depends on larger issues: climate change, ocean acidification, pollution, habitat destruction. These are global problems requiring global solutions. Changes in how we produce energy, how we manage waste, how we regulate industries, how we think about our relationship with the ocean. Sea fans can't speak for themselves, but they're speaking anyway through their slow growth and their ancient existence, testifying to what's possible when organisms are given time and stable conditions, warning us about what we lose when we disrupt those conditions faster than evolution can respond. The water is growing darker now, not because the sun is setting, though perhaps it is somewhere far above, but because you're sinking deeper into rest, into that quiet space where thoughts slow and drift like plankton, where the boundary between waking and sleeping blurs like the boundary between water and sky. The sea fans continue their patient work. They'll be here tomorrow. They'll likely be here years from now, decades from now. If we're careful, if we're thoughtful, their polyps will extend into the current. Their colonies will grow, imperceptibly slow but unstoppable. New larvae will settle. Old colonies will eventually die and be replaced. The forest will persist, changing but continuous, the same and yet different, maintaining its patterns across timescales we can barely comprehend. You've drifted through their world tonight. You've learned something of how they live, how they grow, how they build their elegant structures and create habitat for thousands of other species. You've seen them in shallow tropical waters and in the deep cold darkness. You've understood something of the threats they face and the efforts to protect them. And now it's time to let that knowledge settle, to let it become part of the background of your understanding of the ocean, a quiet awareness that adds depth and richness to your appreciation of the sea. The sea fan's branches sway gently in the current, moving with the water the way you're moving towards sleep, following natural rhythms, responding to forces larger than yourself. Each polyp tends its small territory, extending its tentacles, capturing what it can, sharing with its neighbors. The colony functions as one, thousands of individuals creating something greater than themselves. And you, drifting now, half aware, you're part of something greater too, part of the living world, connected to these corals and all other life through evolutionary history, through shared chemistry, through the simple fact of existence. Let your breathing slow to match the gentle pulse of the current. Let your thoughts spread out like branches into the blue. Let yourself grow heavy and still like the holdfast anchoring the coral to its rock. You are safe. You are at rest. You are held by the ocean, by the darkness, by the slow approach of sleep. The sea fans will be there in the morning. The forest will stand. The polyps will feed. The current will flow. Everything continuing as it has for longer than memory, as it will long into the future. And you can rest now, knowing that somewhere in the ocean, in many oceans, on countless reefs, the slow architects continue their work, building structures of lace and calcium and protein, creating beauty without intention, offering shelter without asking anything in return. Sleep now. The coral forest is watching over the night, and you are part of that night, part of the ocean's long, slow breathing, part of the current that flows through all living things. Rest, sleep, let go. The sea fans sway in darkness, and you sway with them, down into the gentle depths where dreams begin. Good night.